Good afternoon. First of all, I would like to thank the organizing committee for selecting our work for this oral presentation, which title is Lipid peroxidation and protein tyrosine oxidation are mechanistically associated events in hydrophobic biocompartments. So, tyrosine is an aromatic amino acid present in proteins in relative high percentages, and due to its phenol moiety, it has an intermediate value of hydropathy, and therefore it may be present both in solvent-exposed and internal regions of proteins. When tyrosine becomes oxidized by oxidants such as NO2, hydroxyl radical, carbonate radicals, tyrosine radical is formed, and in the presence of NO2, 3-nitrotyrosine will be formed. The phenolic group of tyrosine has a pKa of 10.5, and therefore it will be mostly protonated at physiological pH. However, when tyrosine becomes nitrated, these pKa values drops down, and therefore both phenolate and protonated forms will be present at physiological pHs. Protein tyrosine nitration represents a post-translational modification mediated by nitric oxide derived oxidants such as NO2 and peroxynitrite, and it involves the substitution of a hydrogen by a nitro group in the position 3 of the phenolic ring of tyrosine. Tyrosine nitration may promote dramatic changes in protein structure and function. In the case of function, it may promote either a loss or a gain of function. It may alter phosphorylation cascades, which involve a particular tyrosine residue, or induce immunological responses against the nitrated proteins. It can also, nitrated proteins or oxidized proteins can promote degradation by proteasome or formation of aggregates, as we can see here. The reparation reactions of these products are still under debate, so we don't understand very clearly which is the mechanism if it exists. Finally, 3-nitrotyrosine is a footprint of oxidative stress in vivo and has been revealed as a strong biomarker and predictor of disease onset and progression. So the formation of 3-nitrotyrosine involves the intermediate formation of the tyrosine radical, which, as I say, can be performed by different oxidants. And in addition to 3-nitrotyrosine, other products can be formed, as, for example, the formation of dityrosine, which involves the recombination reaction of two tyrosine radicals. So, tyrosine nitration depends in several factors, which include the protein structure, the nitration mechanism, and the environment where the tyrosine is located. Usually in polar environments, it is enhanced by carbon dioxide and transition metal centers. It is facilitated within proteins which have acidic amino acid nearby the tyrosine residue, loop structures, and binding sites for hemoperoxidases or metals, and it is inhibited by antioxidants that are normally present in the aqueous phase, such as glutathione, which reacts with NO2 and therefore inhibits tyrosine nitration. Most of the mechanistic studies on tyrosine nitration have been performed in aqueous solution. However, the physicochemical factors controlling this process may be different in hydrophobic environments where we have a great amount of unsaturated fatty acids, which may outcompete for the free radical species. We have an exclusion of antioxidants normally present in the aqueous phase, such as glutathione, and we can have an extra diffusion and concentration of species which participate in nitration, such as NO2 and NO. So, in order to study the nitration mechanisms in hydrophobic environments, we developed a series of probes in collaboration with Dr. Balaram and Kalyana Raman, which are BTBE, which is a hydrophobic tyrosine analog, which has a therapeutic moiety which is anchored in the bilayer, and a series of transmembrane containing tyrosine-containing peptides, which have 23 amino acids which occupy the membrane, and in which the tyrosine residue is present at different positions, position 4, 8, and 12. So these are the probes that we will be working on in order to study the nitration mechanisms in membranes. So the experimental strategy was to incorporate these probes, either BTBE or the transmembrane peptides, to liposomes or membranes. These liposomes have different fatty acid compositions, so we will work with saturated and unsaturated liposomes. We will expose these liposomes to oxidants such as peroxynitrite. We perform an organic extraction, and then we will separate the oxidation products by reverse phase HPLC. And here we can see a typical chromatogram where we can show the elution of the nitrated product of BTBE, as we can see here, or the dimer. And also, we performed a similar experiment to the detection of the nitro derivative of the transmembrane peptides. So, if we expose BTBE containing liposomes to peroxynitrite, nitro-BTBE is formed, and the most likely mechanism is by the intermediate formation of the phenoxyl-derived radical, as it happens with tyrosine, and alternatively we can have the formation of the dimer by the combination of two phenoxyl radicals. So we performed EPR spin trapping experiments in order to detect the formation of the tyrosine radical derived from BTBE, and we worked with a spin trap called MNMP, and when we treated the liposomes containing BTBE with peroxynitrate in the presence of the spin trap, we were able to detect a 3-line signal which is in agreement with the formation of a tyrosine radical within the membrane, as it happens with tyrosine in aqueous phases. So when we exposed liposomes, saturated liposomes with BTBE to peroxynitrate, we were able to detect the formation of nitro-BTBE in a dose-dependent manner, and we asked the following question: which would be the mechanism of nitration in unsaturated liposomes, where the important amount of unsaturated fatty acids could outcompete for the free radical species? However, when we performed the same experiments in the presence of unsaturated fatty acids, we saw that nitration yields were higher. So this took us to the idea that maybe lipid peroxidation processes that were taking place within the membrane were fueling somehow the nitration process. We performed the same experiments with the different transmembrane peptides, Y4, Y8, and Y12, and we saw that nitration was dependent on peroxynitrate concentration, and that nitration yields were slightly higher for the peptides also. The initial reactions of lipid peroxidation involve an initiation reaction which involves the oxidation of a fatty acid by oxidants like hydroxyl radical to yield the alkyl radical, and a propagation reaction which involves the addition of oxygen to the alkyl radical in order to form the peroxyl radical. So, if lipid peroxidation reactions are participating somehow on nitration reactions, we should see an effect when the experiments are performed under low oxygen tensions. And indeed, when we performed the nitration of BTBE incorporated to liposomes of different fatty acid composition, we saw that the nitration yields were very lower when the experiments were performed under low oxygen tensions. We performed the same experiments for the peptides, and we can see that, again, when the experiments were performed under low oxygen tensions, the nitration yields were lower. So this is confirming the participation of lipid peroxidation reactions in the nitration process. We also incorporated BTBE to red blood cell membranes, and we were able to demonstrate an effect also of oxygen in a membrane, in a real membrane. As we can see here, the nitration yields were lower in the presence of lower oxygen tensions. So if lipid peroxidation reactions and nitration reactions are taking place at the same time, we should be able to demonstrate the formation of the radicals, the lipid radicals and the phenoxy radicals. So we perform again EPR spin trapping experiments, and we saw that when BTBE was incorporated to unsaturated liposomes, XYLP-C, the signal was much important than when BTBE was not present, and here we can see the signal for the phenoxyl radical alone. So we were able to demonstrate that lipid peroxidation reactions and nitration reaction were taking place at the same time. So what we proposed as our hypothesis was that if peroxynitride-derived radicals are able to promote lipid peroxidation and generate the peroxy radicals in the presence of oxygen, peroxy radicals could be able to oxidize tyrosine to yield a tyrosyl radical, and therefore, in the presence of NO2, allow the formation in the membrane of 3-nitrotyrosine or the dimer, as we can see here. By oxygen consumption experiments, we were able to demonstrate the reaction between tyrosine and lipid peroxyl radicals, with a rate constant of 10 to the fifth. And once we were able to demonstrate this reaction between tyrosine and lipid peroxyl radicals, we asked the question if peroxy radicals and tyrosyl radicals are formed at the same time, probably there would be a radical-radical termination reaction between both, and therefore the formation of adducts between themselves. So we performed the co-oxidation of BTBE in the presence of PLPC, as we can see here, and an azo compound, and we were able to detect the formation of an adduct between both of them. The formation of this adduct involves two main reactions: a para-coupling reaction between the tyrosyl and the peroxyl radicals, and this is followed by an intramolecular cyclization which yields Diels-Alder type products. So I will finish by concluding that we were able to demonstrate a connecting reaction between lipid peroxidation and tyrosine nitration in membranes, which involves the participation of the peroxyl radicals attacking tyrosine and generating the tyrosyl radical. And therefore, we propose that this could lead to the formation of adducts between tyrosyl and lipidic radicals. So our conclusions are that tyrosine nitration in membranes is closely related to lipid peroxidation reactions. We were able to identify this connecting reaction. As I showed you previously, we demonstrate, the formation of adducts between lipid and tyrosyl radicals. And we performed experiments with the transmembrane peptides, as you saw, which recapitulate the data obtained for the BTBE previously. So just to finish, I would like to thank the people involved in this work. From Montevideo, Uruguay, Dr. Rafael Radi, Dr. Madia Trujillo, and Jorge Wenzel. From the Medical College of Wisconsin, Dr. Balaraman Kalyana, Marcos Lopez, and Joy Joseph. And from Vanderbilt University, Ned Porter, Roman Shepin, and Matthias Moller. So thank you very much, and I will be happy to answer your questions.
A few questions. We don't have very much time. Yes, Enrique, please.
Silvina, are we talking peroxynitride or peroxynitrous acids?
Yes.
Which is the species that actually is going through the membrane?
Well, the species that will go through the membrane is the peroxynitrous acid, the protonated form.
Yes.
In the case of liposomes, because you won't have the channels for peroxynitrate anion that you can have, for example, in cells.
And the dissociation of this cage reaction where you have those two radicals in peroxynitrate is the same in the lipophilic medium that is in the...
Yes, once peroxynitrate goes into the membrane, it will homolyze, and you will have the free radicals NO2 and OH, and you will be able to go with the reaction. Indeed, we were able to demonstrate the formation of hydroxy-BTBE, the hydroxylated derivative within the membrane, which confirms that the homolysis is taking place in the membrane.
And do you see any difference when you're using Y8, Y12?
Well, those experiments regarding the yields, yes. What we see, but these are preliminary data, is that it seems to be more nitrated Y8. However, previous data performed by Raman, by Balaraman, Kalyana Raman, are slightly different. So we are just beginning to work with a comparison of the three peptides, and I am in that way. So I can't say you exactly which is the peptide that is being more nitrated. In my experiments performed by me, the nitration yields are higher for the Y8, the intermediate position. Raman saw more nitration in the Y12 when he was working with slow infusion of peroxynitrate. I am mainly working with peroxynitrate as a bolus.
What I like is also that you see this potentiation with the peroxyl radical, yeah?
Yes.
But is it possible that you form also nitrated lipids?
Yes, yes.
That will be a termination reaction.
Also.
Antioxidants.
Yes. You will be forming the nitrating lipids also. Yes. But even having other products that are very important within the membrane, we were able to detect the nitration of, for example, the BTBE or the peptides. So that's why we started thinking that lipid peroxidation reaction were fueling the process and not only competing with the nitration species. Okay, thank you.
Okay, Kevin, please short question and short answer.
I'm going that way.
On Enrique's question, what's the pKa of peroxynitrous acid?
6.9.
So
you will have some,
bit.
and yes.
But a very little bit. Enough to make the reaction work? Because I don't know.
No, you will have both species.
Well,
yes.
I said before you have an equimolar mix, but if it's pKa is that low, below physiological pH, I'm not sure.
With 6.9, you will have some percentage.
Right.
We can make but you will have both species. Yes.
Is it enough? Is the question.
Yes.
I will pass the microphone to Juan.
Just a quick question. Very naive, but let's go back to physiology. If you're correct in saying that at 2% oxygen in your reactions, nitration is significantly low,
Yes.
then my question is about nitration reactions in vivo in the eye, in the spleen, in different vascular beds where oxygen tension clearly varies. And I wondered whether the nitration reactions that you're describing vary and are affected in vivo by the oxygen tension in vivo.
Do you ask if we think that yes? Yes, we do. We do.
Do you have evidence for that?
Not yet. No, not yet.
Because that would be very interesting in terms of the vasculature when we're looking at the pulmonary circulation coming down.
But the results we have obtained with red blood cells are an indication that that will be taking place in cells as well.
Thank you
Okay.
very much.
No, please.